Hi there, I'm Daria and I'll be talking about Hsp90 for the next 5 minutes or so. So Hsp90 stands for heat shock protein 90, which is a heat shock protein with molecular weight 90 kilodaltons. Considering that would be included, it implies the presence of other sized HSPs, which is true. Notable examples would be Hsp70 and Hsp40, and the heat shock in the name means that this protein protects its substrates, or clients, from stress due to elevated temperatures in the cell's environment. A fairly important bit of information would be that Hsp90 is a chaperone protein. In general, chaperones can do a few important things, like assisting proteins in reaching their native conformations, protecting their clients from heat shock during complex assembly, preventing protein aggregation, or mediating targeted unfolding and disassembly. Chaperones can operate on a wide range of structures, mostly due to their lack of a well-defined pocket for client binding. And in this video, I will be talking about Hsp90 structure, cycling mechanism, and clinical applications. If that's not exciting, I don't know what is. So this first slide shows the open conformation of Hsp90 in ribbon form of a previously resolved crystal structure, and as you can see, the protein is a homodimer. Each of the dimers is composed of 3 flexibly joined domains, namely the CTD or C-terminal domain, which is shown in blue, the middle domain, green, and the N-terminal domain, orange. So the N-terminus has a pocket for binding ATP, which is indicated with white arrows, and the amino acids in the pocket directly interacting with ATP are listed on the right. They're the amino acids forming indirect hydrogen bonds through water, except for aspartic acid 79, which also forms a single direct bond with N6 amino group of the adenine base of ATP. Basically, many involved amino acids are polar, uncharged, or negatively charged, depending on what they are interacting with. The N-terminus is connected to the middle domain by a charged linker, which helps with co-chaperone recognition. The middle domain is thought to have an important role in client recognition. Located there are a hydrophobic region as well as an amphipathic projection, which are crucial with interactions with clients. The amino acids involved with that are shown on the side below the ATP binding. The C-terminus is involved with mediating dimerization, and its last 5 C-terminal residues make up a domain binding site that mediates interactions with a variety of co-chaperones. This is a top view of Hsp90 using the same resolved structure. In transition to closed conformation, the N-termini come together in the center, and it's why on the opening slide the open versus closed conformations looked like a twisted clamp. Closed conformation is shown here in ribbon form on JMOL. The colors just show different secondary structural elements, where pink are the alpha helices and the yellow are the beta sheets. The structure was resolved in complex with the co-chaperone SBA1 and ATP, but this image only shows the Hsp90 homodimer. And what's important is that the closing mechanism doesn't act to clamp on its clients, but rather to create a binding surface upon which clients can attach, and client folding can be coupled to ATP hydrolysis occurring at the N-termini. Here's Hsp90's cycling mechanism that involves ATP and a general client. First, ATPs bind the N-termini, and an inactive client binds the middle domains. Hsp90 is still only in a partially closed state since the N domains aren't dimerized yet. Then co-chaperones are recruited, thus causing the N-termini to dimerize and fully clamp in a twisted conformation. This dimerization of the N-termini subsequently hydrolyzes the bound ATPs and activates the client. The N-termini then separate, the ATPs and inorganic phosphates leave, and the cycle can begin again. This next diagram illustrates the differences in the way that clients can interact with Hsp90. So earlier I said that the closed conformation provides a binding site. That isn't always the case. Here that would happen at picture B, where the Hsp90 is in closed form, but binding can also happen while in open conformation as seen in A. It still doesn't clamp down, but considering the difference between that variety in conformations and the symmetrical opening and closing in the blue section on the right, you can see just how flexible, both with substrate types and interactions with co-chaperones, that Hsp90 really is. And finally, on to clinical applications. If you were asleep before, you might want to wake up now because it gets interesting here. I said that Hsp90 has a wide variety of potential clients, and that is pretty relevant with cancer, because Hsp90 regulates activation and maintenance of several regulatory and signaling proteins like these here listed on the slide. They are oncogenic. As it turns out, Hsp90 is found in different states in healthy and cancer cells. You can see that the ideal form for Hsp90 to be in is it being alone and unbound at low ATPase activity, indicating that co-chaperones are only attaching as they're needed for clients to use. However, in cancer cells, cellular stress and rapid dividing ends up creating a super chaperone Hsp90 complex where several unnecessary co-chaperones and clients are binding to Hsp90 and basically turning it into a monster. Hsp90 is then in a state of high ATPase activity and thus disrupts the function and causes misfolding of its bindees. Consequently, it causes a host of problems leading to cancer. With neurodegenerative disorders associated with protein aggregation like Huntington's, Parkinson's, and Alzheimer's, which is what I will be focusing on, the aggregated proteins like amyloid beta peptide or microtubule-associated protein tau are all Hsp90 clients. Hsp90 and, for example, Hsp70 have opposite effects on client stability, which is used to counteract the negative effect of Hsp90 in these diseases. One of the models is shown here, where the inhibition of Hsp90 with, say, small inhibitors activates specific heat shock protein transcription factor, or HSF1, which induces expression of Hsp70 and Hsp40, as well as other chaperones, which in turn promote dissolution and protein degradation of the plaques and tau aggregates. Hsp90 is in the middle of this diagram, and the two columns on either side show possible chaperones and complexes that could be recruited by HSF1 to combat aggregation of tau and amyloid beta. And here are the references I used in case that's required. And in conclusion, Hsp90 is exciting. That is all.